professor and director of the Center for Marine Science and Technology at North Carolina State University, and Janet Nye, associate professor at the University of North Carolina Institute of Marine Sciences. I'm really particularly excited that we have a little bit of a 1-2 punch of inshore, nearshore, and offshore today as well. So without further ado, let me give you quick instructions on questions, and then I'll turn it over to our speakers. So the way we handle questions in these sessions is that you put your questions in the chat to our moderator, Catherine Anardi, who's here as well. And then after the speakers complete their prepared talks, which are in the range of 20 to 25 minutes, then we'll have the Q&A session, and Catherine will unmute you and call on you to ask your question. And if you're unable to unmute or if you're unable to speak for your own question, she will read the question on your behalf. Okay, so we will turn it over to Dave Eggleston from NC State. Thank you.
All right, great, thank you. I appreciate the Seacoast team giving me the chance to speak with you all this morning, and in particular, Marty and Catherine. So let me share my screen and we will get started. Okay, does that look okay? All right, so I'm going to talk about ecological concepts that drive oyster restoration, and think of this as a case study that we're applying to oyster restoration in North Carolina, but the general conceptual approach and framework can be applied to seagrass, salt marsh restoration. We're applying a similar framework actually to deep sea methane seep communities. So be thinking about these other applications as we go through this case study. And I'm going to discuss hard work done by a lot of previous graduate students, in particular, Brandon Puckett and Seth Tierkoff, who were former PhD students. And then in terms of the Seacoast mission, in talking about mitigation and recovery strategies, just wanted to place habitat restoration in the context of these grand challenges and just re-emphasize the fact that a lot of these coastal nursery habitats, salt marsh, mangroves, oyster reefs, serve as important nursery habitats for fishery species. So they are supporting food security. In terms of ecosystem services, they're providing means to mitigate and adapt to climate change, which is illustrated in this middle picture. So we've got this oyster living shoreline helping to reduce erosion of the salt marsh. The salt marsh is, of course, photosynthesizing, so it's capturing CO2, giving off oxygen, trapping sediments, and also serves as a sort of a sponge in terms of helping to desynchronize floodwaters. And then food security, ecosystem services, combining to support sustainable and resilient socioeconomic systems shown on the right, whether that's commercial or recreational fisheries or improvement of water quality. So here's an outline of what I'm going to talk about. In terms of the conceptual approaches, I'm going to briefly touch on 5 marine protected areas, metapopulations and sources versus sinks, functional equivalency, do restored habitats function similar to natural habitats, integrating ecosystem services into restoration efforts, and then using habitat suitability indices as a tool to guide restoration. Then just touch on oyster life history and current restoration in North Carolina, and then we'll apply those concepts and ask and answer 4 questions. So in terms of marine protected areas, there are areas that I think most of us know that are quite often no-take in terms of fishing. And in this particular illustration, being used to preserve the size and age structure of fish. So we're trying to preserve these big fat females that contribute disproportionately to downstream, for example, larval supply. And then these protected areas also can help preserve community structure. In terms of metapopulations and sources versus sinks, I like to use this mountain sheep population and a mountaintop in Colorado is an example of a metapopulation. Over on the right, those little red areas are these relatively isolated subpopulations, and there's very little mixing of these populations except during mating or under certain, say, drought conditions where these animals might start to move out of these subpopulations to feed. And so this collection of interacting populations is the metapopulation, and some populations contribute disproportionately to that overall metapopulation. So those subpopulations have birth rates greater than death rates, and they're referred to as population sources. And we quite often talk about metapopulations in terms of this dominant eigenvalue coming out of a metapopulation model. So if that dominant eigenvalue is greater than or equal to 1, that's a stable or increasing metapopulation. And then some populations only persist due to immigration from these sources. So their death rates are greater than birth rates. Their lambda value is less than 1. And again, they only persist because of immigration from these source populations. In terms of functional equivalency, a key question is, are restored habitats functionally equivalent to natural habitats? And the literature suggests it can be equivalent, or it can take years, tens of years, 100 years to be equivalent. And so think about things such as growth and survival rates. And I don't have time to show you the data, but the demographic rates of restored oyster reefs in North Carolina are functionally equivalent to, say, oysters in Chesapeake Bay or Gulf of Mexico. And then lastly, this conceptual approach of employing habitat suitability indices, which are, I think, again, a powerful tool to address one of the key questions with habitat restoration, and that is where to restore or conserve if you've got limited money and person power. And then secondly, can we optimize for multiple goals, such as metapopulation connectivity or ecosystem services or recreational opportunities. And so these geospatial habitat suitability indices are spatially explicit decision to support tools. The HSIs have been developed to guide location of aquaculture, fishery production, and restoration of oysters. So that brings us to oyster life history. Oysters release egg and sperm in the water column. Those eggs become fertilized and metamorphose into larvae that drift in the water for about 3 weeks, and then settle typically on other oyster reefs or other hard substrate. And they start to form these structurally complex habitats that can support or serve as essential fish habitat. Already mentioned that they can serve as erosion control through the creation of these intertidal berms. And they're also filter feeders, and so they can limit phytoplankton concentrations and help to reduce anoxia and hypoxia. So why oyster restoration? This paper came out by Beckett et al. In 2011 that basically highlighted the global decline of oyster reefs as a function of percent of historic abundance. And so you can see in our part of the world, we're looking at anywhere from 1 to at most 10% of historic abundances. And so in North Carolina, there are 3 general approaches to restoration. One is the creation of these intertidal berms or living shorelines, and quite often that's led by NGOs such as the North Carolina Coastal Federation, working with a lot of volunteers. And then the North Carolina Division of Marine Fisheries has 2 restoration programs. One is the creation of culch reefs, which are basically spreading out shell or other hard materials on shallow estuarine bottom. That material receives natural larval settlement, the oysters grow, and then they become harvested when those oysters reach legal size. The second restoration approach conducted by the Division of Marine Fisheries is the creation of large-scale subtidal reefs and the creation of a network of no-take reserves. And that's shown on this picture on the bottom right. This is a barge that's dumping 150 tons of limestone boulders on the estuarine bottom in Pamlico Sound. And those boulders each form these sort of volcano-shaped mounds, and this would comprise one of their sanctuaries. And so when we started working on this project about 10 to 12 years ago, the Division of Marine Fisheries had about 10 oyster reserves in Pamlico Sound, and they're shown on the right. These squares are certainly not to scale. They're on the order of a quarter to a hectare to maybe 2 hectares. And the distances between these subpopulations ranges from 10 to 125 kilometers. So the sanctuary program goal was to create a self-sustaining network of protected reefs as a means to form sort of a larval insurance policy for the entire oyster metapopulation. And if they didn't have a self-sustaining network, then the key knowledge gap was where best to focus future restoration efforts. And so now we can start to ask and answer those 4 questions. First is what is the relative contribution of each reserve to the network? What is the potential for reserves to persist as a subpopulation connected by dispersal? How does the entire metapopulation interact? Do we see sort of consistent sources? And where should reserves be built, and can we optimize for multiple goals? And so we generated a spatially explicit stage-based matrix population model, had a time step of 2 months to correspond when we were going out in the field and taking measurements of oyster demographic rates. And we initially modeled 10 reserves and 6 size classes, but we expanded that to, so initially it was Brandon Puckett's dissertation that generated this model, and then Seth's dissertation expanded this model to 646 reefs, again, to capture the entire metapopulation. So what I'm illustrating here is basically 1, 2 of the 10 reserves and 1, 2, 3 of 6 size classes. And what we want to do is generate these what are called transition probabilities in the population. So the probability that an oyster remains in a given size class or grows to the next size class, or the potential larval output from a given size class and from a given reserve, and then also the connectivity among this metapopulation. So the probability that these larvae actually recruit back to the natal reserve or seed other reserves. And so we used a variety of field approaches. We used scuba to excavate oysters and get a sense of their basically mark recapture studies that track their growth and survivorship over time. Stripped the eggs out of these oysters, ran them through an optical particle counter to get sort of larval concentrations, and then plugged this into a coupled hydrodynamic and particle tracking model that would estimate larval connectivity. And let me go back. There we go. And just to give you a sense of what larval connectivity looks like in terms of the importance of location. So for example, if we release these virtual larvae from Deep Bay, we see relatively restricted dispersal. And yet, if we release larvae, for example, from Bluff Shoal, we see really high connectivity along the main wind axis of Pamlico Sound. And so now answering these questions, what's the relative contribution of each reserve to the network? And what we ended up through Brandon Puckett's dissertation is something that looks like this, where we can have reserves that can be characterized with oysters as the relatively fast growers, reserves characterized as the relatively high survivors, reserves characterized as the relatively strong spawners, and reserves characterized as relatively strong connectors within the metapopulation. And so if you're a manager faced with this, it's kind of a conundrum on how you pick what to emphasize in terms of restoration. And this is where the strength of this metapopulation approach comes in, because we can integrate all of this information into that single dominant eigenvalue for the overall metapopulation, or generate a patch lambda value, if you will. And basically, that allows us to ask and answer questions like this. What is the relative contribution of each reserve to the network? And what we found was that there were only 2 sources out of 10 reserves and in only 1 of 3 time periods. And so May, June is when we see the dominant spawning event of oysters in North Carolina. There's some trickling of spawning through the summer and then sort of a secondary peak in late August. And so, you know, the question, oh, and then on the legend here, the small circles and cooler colors represent relatively decreasing lambda values, and the big red circles are lambda values that are one or greater. So again, only 2 sources out of 10 reserves, and in only 1 of 3 time periods. And so the question, what is the potential for reserves to persist as a network connected by dispersal, which again was the goal of the division marine fishery? And the answer is that the potential is low to no, except for 8 of these 10 reserves, densities were relatively high and stable. 2 of the reserves had collapsed, but the other reserves are still functioning pretty well 15 to 20 years later. And so it begs the question that this network of marine protected areas is actually being sustained by larval spill-in from fished areas. So then that begged the question, how does this entire metapopulation interact? Are there certain natural fished reefs that are serving as important potential sources? And so again, Seth Tierkoff applied the metapopulation model to 646 reefs, and they ranged from sort of subtidal culture reefs shown in green to hardened shorelines shown in red to natural intertidal reefs shown in sort of light blue, natural reefs shown in purple, and sanctuaries shown in black. And so the next question, are there consistent sources? And what Seth found was that there are actually some consistent source subpopulations in space and time, and they tend to cluster around the northeast and southwest portions of Pamlico Sound, and driven in large part by favorable salinity, favorable larval dispersal and connectivity. And so then the next question is then, where should reserves be built and can we optimize for multiple goals? And so this is the workflow for the habitat suitability tool development. And the first thing to do was to prioritize relevant spatial factors and things like salinity and proximity, for example, to boat ramps that would allow fishermen access to these areas. Bottom type, so the reefs wouldn't sink in the mud. And an important component of prioritizing these factors was to do it with a working group of stakeholders that consisted of fishermen, managers, and academic scientists. Once we had that priority list of spatial factors, it was then time to assemble the spatial data, which was very labor intensive because a lot of this data was buried, sometimes literally in file cabinets. Then to integrate that data into this GIS-based suitability model, did our best to validate the model with the data that was available, and then applied the model to inform restoration. So where should reserves be built? Again, we created this a grid of Pamlico Sound with over 6,000 square kilometer cells, conducted larval dispersal simulations, and assembled 17 GIS layers that were either exclusion layers or threshold layers. So exclusion layers might be things like these military training zones shown in red, which were no-go for restoration. And threshold layers might be things like salinity, where green would be sort of these middle salinity zones given a score of 1. Oysters do quite well there. Areas in red were given a score of 0. These are relatively low salinity areas where oysters don't survive very well. And areas in yellow are relatively high salinity areas. Oysters survive there, but they're also prone to pests and diseases. And again, the working with stakeholders was key to identifying the weighting of these threshold layers. And so doing this, we end up with something like this, where suitable areas are shown in dark green and unsuitable areas are shown in red. And so for example, areas in red right here behind the Outer Banks really was driven by water depth. With these sort of volcano-shaped mounds, we need to have about 2 meters of clearance with the surface of the water so that there's no navigation hazard. So we attempted to validate this model. We mined the Division Marine Fisheries fishery independent data for their density data on these reserves over time. An important thing to realize is when you create these reefs, you'll see pretty rapid increase in oyster density. It'll reach sort of a plateau and then start to level out. And so these reefs were created at different times. And so we attempted to normalize density by reef age, and that's shown on the y-axis here. And then our habitat suitability scores are increasing on the right of the x-axis. And so oyster density increased exponentially with these habitat suitability indices. We then looked at the sensitivity of the model and basically removed certain parameters, reran the model, and then replaced and removed other parameters. And percent change in the HSI indices was greatest for salinity and larval import. And so this tool has actually now been adopted by the Nature Conservancy. It's part of their Restoration Explorer tool, where you can come over here on the left and play with things like the relative importance of salinity, or the effect of natural reefs. And this has also been adopted by NOAA. So lastly, can we now optimize for multiple goals? So we could optimize for water quality ecosystem services, or fisheries production, or enhanced hook and line fishing opportunities, or shoreline erosion control. And we chose to first try to optimize for water quality ecosystem services. And the first thing was just to identify, what spatially explicit data was available, then go through the literature and look at functional relationships. So for example, the relationship between oyster filtration rates and current velocities, or filtration rates and algal particle concentrations. We used chlorophyll A as a proxy for phytoplankton biomass, and generated monthly chlorophyll A maps from 8 years of MARIS satellite data. The water flow was generated from ADCIRC hydrodynamic model in collaboration with Rick Ludick at UNC IMS. And then the DO data was generated from 522 stations sampled over 18 years, and that sampling was done by the Division of Marine Fisheries. And so just to give you a sense of how we were scoring these inputs, this is mean chlorophyll A concentration suitability. And Pamlico Sound is a relatively oligotrophic system in that the chlorophyll A concentrations really never exceeded 100 micrograms per liter. And oysters really don't start to shut down or stop feeding until we reach about 500. And so we scored areas with the highest algal concentrations, highest, and you can see those along the shores where they're receiving nutrient runoff. So if you have high chlorophyll A concentration, you score high. And if you have a low variation around that, that mean concentration, you also score high. And that's the coefficient of variation shown on the right. Bottom left is water flow, and the literature suggests that at about 15 centimeters per second, oysters will stop filtering. And so we're scoring optimal, so you want relatively high flow to provide food particles to these oysters, but not too high the flow. So that's how we've ranked water velocity. And then dissolved oxygen, again, we're scoring areas with normoxic water highest. And so we then end up with something that looks like this, where we're trying to optimize for reef persistence and water quality ecosystem services. So if we're just trying to optimize for water filtration, we might choose areas shown in purple. If we're trying to optimize for just metapopulation persistence, perhaps the areas in brown. And if we wanted to optimize for both metapopulation persistence and water quality, those would be these areas in green in the red circles. So just to wrap up, some take-home messages from this oyster restoration case study is that this metapopulation concept and the spatial habitat suitability indices are powerful tools to integrate data and guide spatial planning. Stakeholder input shape the model development and enhanced buy-in. Integration of ecosystem services allows for win-win and trade-off decisions. And during this whole workflow, we identified data gaps, liberated a lot of data, and then also used it to synthesize data in the literature. For example, these functional relationships between oyster feeding and water quality. So with that, again, thanks for the opportunity. There are a lot of students that contributed to what I just presented, and a lot of different funding sources. And with that, I'll finish up. Thank you.
All right, thanks, David. If you want to unshare your screen, that's probably easiest. Okay, we have a couple questions. Let's start with Reed. Reed, you had two. If you could start with your stakeholder question and unmute yourself.
Yeah, so I'm unmuted. So one of the things, Dave, you mentioned, sorry, Dave, great talk. I appreciate it. Good seeing you. One of the things you mentioned in developing that model was that you had to get stakeholders involved. They helped you sort of, I think, decide what had higher priority when developing that model. And I'm curious sort of what stakeholders were involved. As you mentioned, depending on what your target is and who those stakeholders involved, they might have very different ideas of what's more important. And I'm curious how, who you brought to the table and how that conversation went to get to whatever your final result was.
Sure, yeah, great question. So for example, we had folks from academia. Mike Peeler from when he was at UNCIMS was participating. We had folks from the North Carolina Coastal Reserve, Coastal Federation. We had several commercial fishermen, and then it was probably 3 different staff members with the habitat enhancement group from the Division of Marine Fisheries.
And the conversation was that an easy conversation? Do you think everybody was on the same page when bringing those sorts of stakeholders together?
Yeah, actually, Reed, it was surprisingly, it was a very enjoyable. We did it twice actually, and it was enjoyable in that we had a common goal, so nobody really had a key axe to grind, and it was really just talking about oyster life history, the relative importance of, say, salinity versus larval connectivity versus even bringing in some of the socioeconomic aspects like the proximity. One of the layers was proximity to where you could stage materials to build these reefs. So that was an important component coming from the Division of Marine Fisheries. And then some of the commercial stakeholders were like, hey, what about access by hook and line fishermen to these reefs? And so that brought in distance to boat ramps. So it was enjoyable in that I think we coalesced around salinity fairly quickly and larval connectivity, but also some of these socioeconomic layers that we weren't quite sure how to rank those in terms of weighting.
Thanks.
Okay, a question just came in from Megan that I think is a good follow-up. Megan, can you ask your question? Unmute yourself.
Hi, thanks. I was learning a lot here. And just in how you approach this work, to what extent are you really focused on this individual site versus developing, identifying factors, restoration factors, developing criteria and tools that are generalizable and would apply to other sites? How much is that a motivation? And if it is at all, then what is that class of sites, right, that would fall under the umbrella of something that could be generalized?
Yeah, and that was the point that I was hoping to make at the very start, is that this metapopulation framework is I think a powerful tool, and it can be applied to, you can think about seagrass restoration and looking at seed dispersal, for example, or looking at demographic rates of seagrass as a function of different location and different times. And so, those types of life history parameters integrate into this type of metapopulation framework, which then in turn can feed into these sort of habitat suitability tools. And what's interesting is that it really has helped in terms of some of the data liberation and I think identifying hypothesis testing that needs to take place, for example, with some of the functional relationships with respect to oyster, so filtration rates and current velocities and particle concentrations. And in the process of putting together this model, but also continuing to do field work, what we realize is that hurricanes are having a big impact on sediment transport in Pamlico Sound. I think many of us knew this, but now we're going out to sample reefs that are then buried after, say, Florence or after Hurricane Dorian, and yet we're seeing some of these shallow culch sites, which are relatively unaffected by sediment after these hurricanes. So I think the next step, in my mind, in terms of refining this suitability tool, is to now start to integrate sort of probability of sedimentation based on storm-driven sediment transport.
Okay, I want to transition to talking about eigenvalues. We have two complementary questions about eigenvalues. Brad, do you want to unmute yourself and start with yours?
Yeah, well, first, yeah, great talk. I really learned a lot in a short time. Thank you. And I think some of the answer came in your later slides, but I was wondering, when you just had those two reefs with eigenvalues greater than 1, what are the factors that are most strongly determining whether the population's growing or dying in a particular reef?
Yeah, basically those sort of patch scale lambdas, those lambda C's are really you get benefit by your contribution of larvae to other areas, and then you get penalized by your relative death rates. And so what was driving contribution then, again, that time period was in May, June, when we see the biggest spawning output in this population. So the positive lambda C's in those two cases were driven primarily by a larval output and connectivity to the broader metapopulation in concert with high growth and survival rates.
And what determined the high spawning rates?
In terms of the May-June period, again, these animals are sort of building up their energy reserves during the course of winter and spring. And that's why we tend to see that big spawning pulse where these larvae are being put in the water column, where they can actually feed on algal material in the water column. Now, why those two particular locations have really good survivorship, I think has to do with the fact that it's kind of that moderate salinity. And so they're not as prone to disease and pests in those areas compared to, for example, the two sanctuaries that collapsed were in relatively high salinity areas near Hatteras and Ocracoke.
Thank you.
Okay, I saw that Marty had two questions, and Marty, your second question is really good. So if you could ask that one instead.
Yeah, I agree with you. My second question is better. So, but it relates to my first question and to Brad's question. So Dave, that was really a wonderful talk, and having worked a good deal on bioeconomics of metapopulations, I'm really fascinated by this stuff and the way that you're able to quantify these things in a real system, that has direct policy implications. So my question is how, related to that overarching interest of mine is how do you decompose the effects of human intervention and the effects of the natural system dynamics, such as the hydrodynamics, on what's ultimately resulting in the things that you measure for the metapopulation. So in those things like eigenvalues, how do you really attribute what's purely because of human intervention versus this would have happened anyway in this natural system? And it relates to my first question, which was would you measure things completely differently if you say, we were in an alternative world in which you plop down yet another reserve in the middle of this that maybe reinforced the connectivity in some sense? Would all the numbers change and you'd have a completely different bottom line? So now I've made it a particularly hard question. Over to you, Dave.
All right, so a lot of ways to unpack that. I guess the first thing is, if we think about, let's go back to marine protected areas as a management tool. And I don't know if we have time, I can show you the data, but basically what it shows is that if we're to look at the size frequency of oysters in natural fish reefs, you'll see that there's basically hardly any size frequency at all and hardly any of the large fat females that contribute disproportionately to reproduction. And in contrast, if you look at the size frequency in these marine protected areas, we see a very, a nice natural size structure, a lot of juveniles, but we still see a lot of big fat females beyond that legal size class. And when you look at their potential larval contribution to the overall metapopulation, it is almost, it's 40% or higher. And they have a very small footprint compared to the footprint of natural reefs. And so from a human intervention standpoint, we're getting a lot of bang for the buck out of these no-take reserves in terms of potential spawning output. So that's I feel like the human intervention part of this. On how that's coupled with nature, I think also has to do with the fact that this is not a self-sustaining network of reserves, and so that the populations within those reserves are being sustained by the wild population. And so then that puts the spotlight back on how humans are managing those wild populations, because they are contributing to this sort of larval insurance policy. So I think, the key is making sure that we're managing both in tandem. And I think that it further goes back to this idea of functional equivalency, where, again, when you look at the broad literature, and I'm including terrestrial examples, it can take a long time to reach equivalent underground or above ground biomass, production estimates with grasses or what have you. And so the take home message there quite often is conserve rather than restore. So I think there are a lot of different, facets to this that couple humans and natural processes. Did that answer your question, Marty?
Thanks. Yeah. And there's, of course, much more to talk about. But I think Catherine's going to tell us we need to move on in order to get to our next speaker.
All right, Janet, you're on. Thank you so much, David.
Sure. Thank you.
Yeah, that was great. I really enjoyed that. Seeing Brandon's work and how it has expanded. Okay. Are you, there we go. You see my title slide?
Looks great.
Okay. So I'm gonna talk about, first of all, thanks for inviting me to speak. I'm a new associate professor here at the Institute of Marine Science at Chapel Hill, and I am been working in the Northeast US for the last 10 or so years. So I've picked out a few examples of some of the changes that we've already seen happen to Northeast US fish in response to climate change, to give you an idea of the kinds of things that I hope to do now in the Southeast US. So as you all probably know, as waters warm, the great thing about fish and other mobile species is that we think they can adapt. And the first sort of hypothesized response and what we've seen globally is that indeed these fish species have moved poleward as waters have warmed. And so in 2009, we published a paper in which we looked at 36 different fish stocks in the Northeast US. Of those 36 fish stocks, 24 of those had exhibited shifts in distribution that were consistent with climate change, so either moving poleward or to deeper depths. And this is just one example showing the center of biomass. And the red, these are the slopes that we saw in the shift in the center of biomass. 17 of the 36 stocks exhibited a northward shift, which is what you would expect with climate change, and only 4 exhibited a statistically significant southward shift in abundance. And so since that time, I've been really trying to understand the mechanisms behind those shifts. At the time, it was just really important to document that these shifts were already happening. And then I've spent the last 10 years trying to figure out the mechanisms, in particular how those relate to population dynamics and the stock assessments by which we set harvest limits for fish. And so what I really see is two dominant mechanisms that have important implications for stock assessment and management. And the first is probably the one that's most intuitive to people, is that fish are on the move. They are, individual fish are physically moving from areas that are perhaps poor habitat or temperature, suboptimal temperature, to waters that have more favorable conditions. And the second mechanism is that population productivity is changing spatially. So this is a population level process by which the northern populations tend to have more favorable environmental conditions. Those populations are increasing in abundance, whereas in the southern or equatorward range limit, those populations are not experiencing the same favorable conditions, and you kind of have the seesaw effect where the northern population is increasing in abundance. Some people have called that an apparent shift in distribution. It's still a shift in distribution that fishermen and managers have to adapt to. But again, I've been focusing mainly on how this affects the total biomass that we can harvest for these fish species. So if the fish are on the move, you may have the same amount of fish, which means your harvest limits don't have to change, but there are still important management implications. Fishermen might have to move greater distances to catch those fish species, and there are some very contentious issues with allocation between states. For the second mechanism, and this is the one that I think is what we predominantly see in the Northeast US, is that you do have these changes in population productivity, which means that you do have to alter your harvest limits. And that might mean that the northern stock or subpopulation, you could fish a little bit harder, but the southern stocks you really need to reduce fishing, and importantly, adjust your population models to accurately determine those harvest limits. So I'm going to give you two examples of those particular mechanisms, that also gives you kind of a flavor of the kinds of things that I do. I'm going to start with silver hake. This is a gadded species related to cod. It's not as commercially important or as heavily fished as cod, but it is a commercially important fish, and it undergoes a seasonal migration northward in the summer and southward in the winter, like many of the fish species in this region. And I'll just go show you a movie from NOAA showing you the distribution of silver hake. So you can see in the 70s and 80s, they were found as far south as North Carolina occasionally, high abundances in the southern New England, New York area, but then you can see they really become dominant in the Gulf of Maine in the more contemporary time period. So that gives you a flavor of the patterns, that overall pattern that we're seeing. And we were able to relate this to the position of the north wall of the Gulf Stream, so to the oceanography of the region. The Gulf Stream north wall is, I believe, the best indicator of environmental conditions, particularly temperature on the shelf. So when the Gulf Stream is in a more southerly position, you have more of this Labrador slope water, which is cold and fresh. The waters on the shelf are colder, and importantly in the Gulf of Maine, you've got cold bottom water coming in through the Northeast Channel. When the Gulf Stream is in a more northerly position, you see more of this warm slope water that's influenced by the Gulf Stream. The temperatures on the shelf are warmer, and the temperature of the bottom water in the Gulf of Maine is also warm. And so we were able to relate this to the distribution of silver hake. You can immediately notice the difference between when the Gulf Stream is in a more southerly position. These color contours are temperature. bottom temperature. And you can see the blue areas are temperatures less than 6 degrees. And silverhakers are really kind of avoiding those cool temperatures. However, when the Gulf Stream is in a more northerly position, you can really see the temperature is much warmer on the shelf. And the red dots are the silverhake biomass. And you can see they're more offshore and more southerly distributed, whereas when it's warmer on the shelf, they are primarily in the Gulf of Maine. And so we related this to the Gulf Stream index here on the x-axis. This is temperature. And so again, as the Gulf Stream is pushed further north, the temperature in the Gulf of Maine or the northern region is warmer, increases. It does so in the south as well, but what doesn't change is the temperature of occurrence that we find silverhake. So silverhake is moving for sure, as you can see from those maps, but the temperature at which they are found and which they prefer does not change. And so, you know, they're moving into this area that becomes closer to their temperature range in the north versus the south. So this is a real sort of clear example of that first mechanism, where this is just the individual fish moving, or the population moving. The second mechanism, again, was changes in population productivity. I'm going to use black sea bass as an example. This is a fish species found in North Carolina, but also it's the most northerly distributed saranid or grouper species in the Northeast US. And so the first thing that we do in the Northeast when you start studying a new species is go to Bigelow and Schroeder's Gulf of Maine, Fishes of the Gulf of Maine book, in which it tells you all about the life history and habits of this species. And I found it very interesting that Bigelow and Schroeder, the experts at the time, wrote, "There's no reason to suppose that they," meaning black sea bass, "would ever succeed in reproducing themselves in the Gulf or in establishing a temporary foothold, even if the rare migrants should spawn there." So they talk about finding these, you know, stray, lost black sea bass every once in a while in the Gulf of Maine, but they would never establish a population there. And they even went on to say that they're just not abundant enough to be an important fishery in the Gulf of Maine, even though it's an important fishery south of Cape Cod. So I'd like to pause right there and just remind ourselves that the experts at the time in the 1950s could never have imagined that there would be a fishery for black sea bass in the Gulf of Maine. It is kind of humbling. And so, as you might imagine, the movie I'm going to show you shows the distribution of black sea bass, more dominant in the southern part of its range over time. And if you kind of look here in the Gulf of Maine, you see a little population pop up every once in a while, and then it just kind of dies back down until you get to the 2000s, late 2000s, and you kind of see a more persistent population there. And so if you look at the juveniles, that was sort of mostly adult signal, but if you look at the juveniles only, this very few juveniles in the Gulf of Maine, and that suggests the lack of a spawning population there. But then if you look at the years after 2003, there's lots and lots of juvenile fish there. So it does seem that they are establishing a population, and the fishermen certainly are finding them in their lobster traps and thinking about how can we take advantage of this new species. So we hypothesized that overwintering mortality was an important mechanism by which they were able to expand their range as temperatures become more mild. We went into the lab and did a lot of physiological studies on black sea bass and developed an overwintering model for black sea bass, and then combined it with this regional ocean modeling system led by Enrique Kirchester at Rutgers. And so this is a really, you can get high resolution bottom temperature and salinity, and we were able to get that for the time period 1959 to 2007. And we combine that with our overwintering model to get maps that look like this. So this is 2003, which was the coldest year for that time period for which we have hindcasts. And our model shows the area of good overwintering habitat, which is shown in red. That means like 90 to 100% of the black sea bass juveniles in this region would survive the winter. But these blue areas indicate basically there will be 0% survivorship. So you can see that even in the coldest year, there is plenty of good overwintering habitat, sort of from Delaware Bay and south. But north of that, the fish really have to move offshore to get into good overwintering habitat. Also interesting to me is that even in the coldest year, there's some good overwintering habitat in the Gulf of Maine. There's no population there, and they probably wouldn't find this little pocket of good overwintering habitat. And even in the historic time period, this is the warmest year now for those 50 years that we have hindcasts, and you can see in the Gulf of Maine, and really the whole shelf is pretty good habitat for black sea bass. So then we went one step further and basically counted up all those red cells from those maps and said, okay, this is the amount of overwintering habitat in each year. How does that relate to recruitment or, the overwintering survival of these juvenile black sea bass? To do that, we related that measure to the surveys that each state has for juvenile recruitment indices for lots of different fish species. And we found what you might suspect is, and that is a positive correlation between good overwintering habitat, the amount of good overwintering habitat, and the amount of black sea bass recruitment that following year. But you don't see that in New Jersey and South because there's always good overwintering habitat there. Interestingly enough, there's no, or maybe a negative relationship between this index and the coastal survey, but that's kind of the subject of a different talk. And then what we did for black sea bass, similar to what I've done for some other species, is then project further into time and ask how overwintering habitat might change by the end of the century. And so basically this is showing you the relative change. And these red areas indicate, a 90 to 100% increase in black sea bass overwintering. So basically, almost the entire shelf is going to be, good overwintering habitat for black sea bass by the end of the century. And there should be no problem for them to establish a further population there. Now I want to switch gears a little bit and talk about ocean heat waves and switch from the single species work that I've been doing to more ecosystem modeling. So in 2012, the Northeast US and really the whole Northwest Atlantic experienced a really intense and the largest marine heat wave in the Atlantic ever. And we said, hey, this could be a window into how coupled natural systems might change in the Gulf of Maine. We saw some really strange things happen and some really crazy things with the lobster population happen. And so I'm not going to talk about that work, unfortunately, but what happened was, this is that 2012 warming event, and I really expected that that was maybe a one-off and that temperatures, would come back down as they sort of always have in this time series. What has happened is that temperature has stayed really, really warm. And in fact, we've had, two more heat waves since that time. So we thought that 2012 would be a window into climate change when in fact, it seems to have persisted. And instead of kind of looking at how that affects individual species and the fisheries, we really turned our focus to how this affects the ecosystem as a whole. And so these figures here, don't worry about the individual species, but suffice it to say that this is a food web model for the Gulf of Maine. And now what we're really looking at is how do the combined effects of what we see in all these individual species, how do those affect, one, the energy flow in the food webs, two, the total fisheries productivity and catch. So our, like all of our fisheries productivity gonna just systematically go down. And then ultimately ecosystem stability and resilience. So, one strategy for climate change is to just have sort of resilient populations and fisheries, and so we want to also see that in the ecosystem. And so what's predicted with climate change is that we will see, one predicted response in the food web is that you might see a switch from large zooplankton to small zooplankton, and you can see that if this that happens, a lot of if you follow these colored arrows, you can see where that energy goes in the food web. And so you see a lot of that energy going in this particular food web to the detritus, and less going up to these upper trophic levels, which are our fisheries. And then in this bottom panel, what this is showing an increase in the large zooplankton. When you have an ecosystem that has more large zooplankton, you tend to see what these red arrows are showing, and that energy flowing up to the upper trophic levels. And then we have several metrics looking at, okay, how does this affect ecosystem maturity and stability, which are measures of how resistant and resilient the ecosystem is to change. And this is just an example of what they've seen in the Baltic Sea as a result of the combination of fishing, climate, and hypoxia. So that's what's coming in the future for the Northeast and maybe for the Southeast as well. Another thing just to mention quickly is I've been working with some social scientists at NOAA who developed some social indicators for a lot of different communities around all of the United States. And this is just one such indicator, a poverty index for all of the coastal communities on the Northeast coast. And I really want to combine this with my interest in the oceanography and marine heat waves. Let's do this. These are showing you all the communities for which they have data. I'm going to just take out the low poverty index or green dots to show you where the most vulnerable or the communities with the highest poverty indices are. And of course, those are in their southern regions. And then what I'm overlaid that with is the duration of marine heat waves. So this is the number of days that heat waves persist. And you can see that in the Gulf of Maine, we have these really long persistent heat waves, but not so much in the South. So if you think about vulnerability as the product of exposure to a heat wave and then this sort of adaptability index, maybe these populations these communities won't be so vulnerable. However, when you look at the intensity of heat waves, so that's like how much the temperature increases, they're much more intense in these southern regions. So I'm interested in kind of looking at this and how adaptable and resilient some of these populations might be. And that I will just acknowledge my students that have done all the work and take your questions.
All right, thanks, Janet. If you could stop sharing your screen.
Yeah.
Perfect. Okay. We have a couple questions in the chat. Let's start with Dana. Dana, can you unmute yourself? No, she cannot. Oh, I know why. I need to let you unmute. Okay. Sorry about that.
It's nice to meet you, Dana.
Okay, Dana.
Yes, thanks for that. Janet, it was a really nice talk. I had a question that you sort of started to answer, but I'm still going to ask it anyway. So in terrestrial systems, we think about biogeographic barriers, such as mountain ranges, limiting poleward migrations. Are there factors that limit kind of poleward migrations in these fish, so things like habitat? And you started to talk about the zooplankton, but I'm curious if you had other thoughts.
Yeah, I've been thinking a lot about biogeographic barriers since moving to North Carolina, and that barrier at Cape Hatteras, which is one of the strongest biogeographic barriers. In the Northeast U.S., there is a bit of a biogeographic barrier between at Cape Cod, between Georges Bank and South, which is much different ecosystem. It's shallow. There's maybe stronger benthic pelagic coupling, but and the Gulf of Maine is really deep. But we have looked at the species assemblages in the Gulf of Maine compared to those other regions, and we do see those mid-Atlantic species going into the Gulf of Maine. So I feel like it's not as strong a barrier as the one in Cape Hatteras, and I think the reason the one at Cape Hatteras is so strong is because the winter temperatures are so different between south of Cape Hatteras and north of Cape Hatteras.
Okay, next question. Let's go with Marty.
Janet, thanks for really an outstanding, informative talk. And I'm going to ask you a question I will push you a little outside of ecology, but I feel like it's a hugely important question for what Seacoast aspires to, which is to really reflect on the question of which institutions and which policy levers might be maladaptive or adaptive to climate change to facilitate more resilience in coastal communities. And I know from the end of your talk that this is something really important to you. So my question more specifically is thinking about these two mechanisms that you've documented, fish on the move and productivity changes. To what extent is the way U.S. fishery management at the federal level does business, to what extent is that adaptive or maladaptive? And I'm thinking specifically of breaking up this system into regional fishery management councils that have jurisdiction over certain stocks, and I understand I'm putting you on the spot. So if you don't want to answer that exactly or dance around it, that's totally fine, because it's very politically contentious. But I want to know what an expert like you thinks about how maladaptive or not this system is.
Yeah, so I was on a sabbatical in Australia for a year, and they do fisheries management a lot different in the Southern Hemisphere. They have a lot of ITQs, and when we talked about adaptation of fisheries management, they're not, it's not even this jurisdiction, the boundary issue is not even an issue down there. And so I feel like, I don't know if there are any managers on the call, but we're putting a lot of time and energy into allocation issues and coordination among councils, and I've thought about that a lot, but there might be another solution. I'll just leave it there.
Okay, thanks for that question, Marty. Reed.
Hey, Janet, thank you for a good talk. And this sort of, I guess, is maybe a more basic way of asking the same question. I'm not sure. So, are states and feds and these different groups thinking about how these fish are moving, how they're likely to migrate, and how they will likely need to change rules, policies to deal with it, right? So are they aware, and are they actually starting to think about how they will change and implement what's needed?
Absolutely. I mean, I've spoken with many individuals, particularly on the Mid-Atlantic Council and the South Atlantic Council, about these issues. Surprisingly less so in the North New England Council, but they definitely are thinking about it, and I think that's really good. They are talking amongst each other and having sort of joint meetings where they talk about this and how we're going to manage that. I mean, we've also had managers from the councils sort of help direct the research so we can give them what they need. And what I find is they're kind of stuck. They're stuck with the law. They don't have a lot of ways to adapt, I guess, their management right now. So they are trying to figure out for sure how to do this. I would say some councils are confronting allocation issues more directly than others.
Okay, with that, that is all of our questions for today. So I want to thank both of our speakers. And then I need to share, let's see, let me find it, slide for next week. Okay, so next week we will have Megan Mullen, Andy Keeler, and Phil Burke presenting on political science, public policy, and planning in coastal communities. Actually, it's not next week, it's 2 weeks from now. So hopefully we will see you on October 22nd, same time. Thanks for joining.
Thanks, everyone.
Thank you.
Thank you.
All right, I'm gonna stop recording. I always forget to do that.